Hello, biology students, and today I'm going to be doing another text audio edition of our biochemistry unit. So today I'm going to be reading over your section 2.3 on carbon compounds. So make sure you are filling out that section 2.3 reading guide that goes along with this, that worksheet that will go right along with this text. All right, let's go ahead and get started. 2.3 Carbon compounds. Until the early 1800s, many chemists thought that the compounds created by organisms, organic compounds, were distinctly different from compounds in non-living things. In 1828, a German chemist was able to synthesize the organic compound urea from a mineral called ammonium cyanate. Chemists soon realized that the principles governing the chemistry of non-living things could be applied to living things. Scientists still use the term organic chemistry, but now it describes something a little different. Today, organic chemistry is the study of all compounds that contain bonds between carbon atoms. The chemistry of carbon. Is carbon so interesting that a whole branch of chemistry should be set aside just to study carbon compounds? It is indeed, for two reasons. First, carbon atoms have 4 valence electrons. Each electron can join with an electron from another atom to form a strong covalent bond. Carbon can bond with many elements, including hydrogen, oxygen, phosphorus, sulfur, and nitrogen. Even more important, a carbon atom can bond to other carbon atoms, which gives carbon the ability to form chains that are almost unlimited in length. These carbon-carbon bonds can be single, double, or triple covalent bonds. Chains of carbon atoms can even close upon themselves to form rings, as shown in figure 2.11. Carbon has the ability to form millions of different large and complex structures. No other element even comes close to matching carbon's versatility. Figure 2.11. Carbon can form single, double, or triple bonds with other carbon atoms. Each line between atoms in a molecular drawing represents one covalent bond. Macromolecules. Many of the molecules in living cells are so large that they are known as macromolecules, which means giant molecules. Macromolecules are made from thousands or even hundreds of thousands of smaller molecules. Macromolecules are formed by a process known as polymerization, in which large compounds are built by joining smaller ones together. The smaller units or monomers join together to form polymers. The monomers in a polymer may be identical, like the links on a metal watch band, or the monomers may be different, like the beads in a multicolored necklace. Figure 2.12 illustrates the formation of a polymer from more than one type of monomer. It would be difficult to study the millions of organic compounds if they were not classified into groups. 4 groups of organic compounds found in living things are carbohydrates, lipids, nucleic acids, and proteins. Sometimes these organic compounds are referred to as biomolecules. As you read about these molecules, compare their structures and functions. Figure 2.12. When small molecules called monomers join together, they form polymers or large molecules. Carbohydrates. Carbohydrates are compounds made up of carbon, hydrogen, and oxygen atoms, usually in a ratio of 1 to 2 to 1. Living things use carbohydrates as their main source of energy. Plants and some animals also use carbohydrates for structural purposes. The breakdown of sugars, such as glucose, supplies immediate energy for all cell activities. Living things store extra sugar as complex carbohydrates known as starches. As shown in figure 2.13, the monomers in starch polymers are sugar molecules. Figure 2.13, starches and sugars are examples of carbohydrates that are used by living things as a source of energy. The chef shown here is drying pasta, which is made principally of starch. Starches form when sugars join together in a long chain. Single sugar molecules are also called monosaccharides. Besides glucose, monosaccharides include galactose, which is a component of milk, and fructose, which is found in many fruits. The large macromolecules formed by monosaccharides are known as polysaccharides. Many animals store excess water in a polysaccharide called glycogen, or animal starch. When the level of glucose in your blood runs low, glycogen is released from your liver. The glycogen stored in your muscles supplies the energy for muscle contraction and thus for movement. Plants use a slightly different polysaccharide called plant starch to store excess sugar. Plants also make another important polysaccharide called cellulose. Tough, flexible cellulose fibers give plants much of their strength and rigidity. Cellulose is the major component of both wood and paper. So you are actually looking at cellulose as you read these words. Lipids. Lipids are a large and varied group of biological molecules that are generally not soluble in water. Lipids are made mostly from carbon and hydrogen atoms. The common categories of lipids are fats, oils, and waxes. Lipids can be used to store energy. Some lipids are important parts of biological membranes and waterproof coverings. Steroids are lipids as well. Many steroids serve as chemical messengers. Many lipids are formed when a glycerol molecule combines with compounds called fatty acids, as shown in figure 2.14. If each carbon atom in a lipid's fatty acid chains is joined to another carbon atom by a single bond, the lipid is said to be saturated. The term saturated is used because the fatty acids contain the maximum possible number of hydrogen atoms. If there is at least one carbon-to-carbon double bond in a fatty acid, the fatty acid is said to be unsaturated. Lipids whose fatty acids contain more than one double bond are said to be polyunsaturated. If the terms saturated and polyunsaturated seem familiar, you have probably seen them on food package labels. Lipids such as olive oil, which contains unsaturated fatty acids, tend to be liquid at room temperature. Cooking oils such as corn oil, sesame oil, canola oil, and peanut oil contain polyunsaturated lipids. Figure 2.15, nucleic acids store and transmit genetic information. The monomers that make up a nucleic acid are nucleotides. Each nucleotide has a 5-carbon sugar, a phosphate group, and a nitrogenous base. Nucleic acids. Nucleic acids are macromolecules containing hydrogen, oxygen, nitrogen, carbon, and phosphorus. Nucleic acids are polymers assembled from individual monomers known as nucleotides. Nucleotides consist of 3 parts: a 5-carbon sugar, a phosphate group, and a nitrogenous base, as shown in figure 2.15. Individual nucleotides can be joined by covalent bonds to form a polynucleotide, or nucleic acid. Nucleic acids store and transmit hereditary or genetic information. There are 2 kinds of nucleic acids: ribonucleic acid, or RNA, and deoxyribonucleic acid, or DNA. As their names indicate, RNA contains the sugar ribose, and DNA contains the sugar deoxyribose. Proteins. Proteins are macromolecules that contain nitrogen as well as carbon, hydrogen, and oxygen. Proteins are polymers of molecules called amino acids. Amino acids are compounds with an amino group, or NH2, on one end, and a carboxyl group, or COOH, on the other end. Figure 2.16 shows one reason why proteins are among the most diverse macromolecules. More than 20 different amino acids are found in nature. All amino acids are identical in the regions where they may be joined together by covalent bonds. This uniformity allows any amino acid to be joined to any other amino acid by bonding an amino group to a carboxyl group. Figure 2.16. Amino acids are the monomers of proteins. All amino acids have an amino group at one end and a carboxyl group at the other end. What distinguishes one amino acid from another is the R group section of the molecule. Figure 2.17. Proteins help to carry out chemical reactions, transport small molecules in and out of cells, and fight diseases. Proteins are made up of chains of amino acids folded into complex structures. The portion of each amino acid that is different is a side chain called an R group. Some R groups are acidic, and some R groups are basic. Some are polar, and some are nonpolar. Some even contain carbon rings. The instructions for arranging amino acids into many different proteins are stored in DNA. Each protein has a specific role. Some proteins control the rate of reactions and regulate cell processes. Some are used to form bones and muscles. Others transport substances into and out of cells or help to fight disease. Proteins can have up to 4 levels of organization. The first level is the sequence of amino acids in a protein chain. Second, the amino acids within a chain can be twisted or folded. Third, the chain itself is folded. If a protein has more than one chain, each chain has a specific arrangement in space, as shown by the red and blue structures in Figure 2.17. Van der Waals forces and hydrogen bonds help maintain a protein's shape. In the next section, you will learn why a protein's shape is so important. All right, everybody, that concludes our section 2.3. Fill out that reading guide, turn that in, and have a wonderful day.